To round things off for the lymphatics and immunity chapter, we need to look into antibodies. Now, there are 5 main categories of antibodies we're focused on. We're going to start with this one, IgM, as the first category. Now, IgM is going to be the first antibody released primarily. So this is the one that really can help out and get there initially before anything else. It is also a potent agglutinator. What that means is it causes clumping, agglutination, clumping. And it also helps out with the complement protein. Now, we discussed complement protein and complement fixing in a previous video. The idea there is complement is going to help with destroying cells by creating openings in them and therefore disrupting and causing lysis. But this is IgM, so agglutinator, complement protein, and first to respond. One thing that's interesting is look at the number of binding sites. Every green piece there is a binding site. So it's going to have more binding sites than any of the other categories we'll talk about. IgA. Now this one is primarily found as a barrier. Think mucuses. It's found in mucus trying to prevent entry into the body. So it's really trying to stop those pathogens from getting through your nasal membrane, through your mucous membranes of your GI tract. It's all about trying to prevent entry initially. So you can kind of see an example here. We're now down in the intestines because these are going to be Peyer's patches. And those Peyer's patches, you can see, have T cells and B cells. So if we're taking this arrow over here and looking at this whole piece, looking at the T cells and B cells are receiving. They're receiving all of the antigens that are being found. What that means is they're getting ready to make these antibodies. If you notice, then go up to the plasma cell, it's making the antibodies, and then they're out here. IgA is out in the secretions, blocking entry of that antigen that was just found back over here. So you're seeing that you have it being found, processed, and then secreted to try to prevent further entry at that point. So all about the IgA antibodies. IgD antibodies. Now, IgD antibodies are the receptors of B cells. That's their primary role. They're on the B cells. These are the receptors. This is what allows the plasma cells to go forth. So you have the B cell receptors are IgD. Then you have IgG. Now, IgG is the majority of the antibodies you find in the blood. I have down here 75 to 85%. That's also why, if you ever see an antibody drawn, it's usually drawn that Y shape, because this is the most common shape because it's 3/4, if not more, of the antibodies you find in your blood. You'll find these primarily as late first response and then secondary responses. So from the secondary responses and late primary response for immunological. An interesting note here is this IgG antibody class can cross the placenta. So these can go into the developing fetus, which is important to make sure that these don't attack the fetus then. That's why it's so important to make sure that everything is meshing, everything's working properly. So IgG antibodies. And then we have IgE antibodies. IgE antibodies. Now these are looking at parasitic infections and some allergies. So they're basically causing reaction in your allergies and parasites. They're going to secrete and cause the mast cells as well as the basophils to release histamine. Remember, histamine is what causes inflammation. So this is triggering your inflammatory response, which makes sense if these are done with allergies. Allergies can cause inflammation, irritation. So this is IgE now. So if you look at all 5 of these together, please do not memorize this whole chart. You can look at it as kind of a reference, but the interesting notes. Look at the number of binding sites. There are more binding sites, obviously, in IgM than anywhere else. Other interesting note, crossing the placenta. The only one that crossed the placenta is IgG. The other 4 can't cross the placenta. And then we know again that we said that the B cell receptor is IgD. So few interesting notes to kind of pay attention to and kind of make sure you remember those highlights. So to finish things off, how does all this work? How does it all come together? Well, we have the different antibody classes. They all have their own functions. But these antibodies, when the antibodies bind the antigens, first of all, neutralization. If you have all these antibodies surrounding the pathogen, can the pathogen do anything? No. It's blocked. It can't bind, can't go here. It's completely surrounded and non-functional. We have agglutination. That's clumping. So if you start clumping everything together, again, it can't function. So agglutination or clumping stops it or inactivates it. We have precipitation. That's causing all the molecules to get stuck together and just fall out of solution. If you precipitate out of solution, it's again not functional. It's not going to do its job. Those are 3 different ways that antibodies can inactivate what they're targeting. You also have, again, the complement here. Remember, you can activate complement by fixing complement, causing that bore of the hole, and therefore lysis of that cell. And that leads to, as mentioned, cell lysis. But you also have here inflammation. Remember, we talked about, again, the causing of histamine release, inflammation, and enhancing phagocytosis. Phagocytosis, the eating or destroying of foreign pathogens, or for that matter, cell debris or anything that shouldn't be there. So really, these antibodies can do a whole lot, causing a whole lot of different processes to occur, all meant to stop that pathogen or to prevent further infection. So a lot of things happening with antibodies when they form that antibody-antigen complex across those 5 different categories of antigens.